Ammonia emissions in the air is an environmental and human health concern. Ammonia can cause chest tightness in low concentrations and cancer or death in high concentrations. Almost 50% of all global emissions are from agricultural sources. While OSHA has some regulations on how much ammonium can be released for a certain amount of time, the limit is usually exceeded on farms. Our experiment will determine ammonia levels on the campus farm, as well as in 2 bathrooms to help determine a baseline.
There were 2 main components to our methodology: sampling and lab analysis. To begin sampling, we first determined sample locations. At each sample location, we set up 3 collection devices. Each device required the following: wearing gloves throughout, we opened the radialo tube, then placed the blue diffusive body on top of the tube and held them together. Turning the tube and diffusive body upside down, the cartridge emptied into the diffusive body. Then a triangular supporting plate was twisted onto the diffusive body and the time of sample placement was recorded. Each empty radialo tube and each diffusive body was labeled with the sample number and location. Once all 3 samples were ready, they were hung in the desired location. Some were not easily accessible, as the sample is severely affected if it is touched. For each sample location, a blank was also collected. After 72 hours, we returned to pick up the samples. Wearing gloves throughout, we cut down the samples and prepared to empty the diffusive body. We had the corresponding empty tube open in one hand and the diffuser body triangular plate in the other, with a triangular plate on top. The plate was unscrewed and the tube was placed on top of the diffuser. Holding it together, then turning it upside down, the cartridge emptied into the tube. The tube was capped and the time of sample collection was recorded. The process was repeated for each sample. The samples were then taken to the USDA lab for analysis. In the lab, we first created the reactives needed, including the buffer solution, phenol, cyanoferrate, and the oxidizing solution. We also needed to create a standard curve to determine ammonium concentrations, so we prepared concentrations of 0.05, 0.1, 0.2, 0.5, 0.8, 1, 2, 5, 8, and 10 ppm of ammonium. Next, we prepared our samples from the field. To get the ammonium collected into water, we opened the radialo tube and added 10 milliliters of deionized water to the cartridge in the tube. Recapped the tube and stirred vigorously in a vortex stir for a minute. Then we transferred 1 milliliter of the solution into a glass vial and repeated this for each sample using a different pipette tip for each. Then we added the reactive solutions to our samples and to our standardized ammonium samples. This included 0.4 milliliters of cyanoferrate, 5 milliliters of buffer solution, and 1 milliliter of oxidizing solution. The glass tubes were then placed under the fume hood for 1 hour. Following the hour, we measured the absorbance for each sample. To do so, we zeroed the spectrometer using the blank corresponding to the sample being tested, filled the glass vial 3/4 full with the solution, and placed the vial into the spectrometer. We tested 6 vials at a time. We ran the spectrometer at 635 nanometers to measure the absorbance of the solution. 3 trials were performed to ensure accuracy.
To calculate the results using the 3 trials in the spectrometer, we took the average of each sample's absorbance and calculated the concentration in ppm using the standard curve. Then, the mass on the cartridge was determined based on the sample's concentration and corrected for each location's blank. From here, using the mass on the cartridge and the exposure time, we calculated the average air concentration during sample collection.
After collecting and analyzing the data, we were able to conclude that the barn had the highest ammonia content and the lowest ammonia content was outside the barn and at the creek. This makes sense because ammonia disperses really fast, so directly outside the barn it got extremely low and the creek got even lower. Inside the buildings it's significantly higher, with the boys' bathroom and bedroom being higher than the girls'. This could mean that there is probably poor ventilation inside the buildings that prevents the ammonia from properly dispersing.